using as well. I try to be brief, but at the end I'll try to make some suggestions what I think can be improved towards including maybe deep learning to make the predictions better. Okay, now I'm trying to use Shiji's. Okay, now, so our methodological workflow is quite similar to what other speakers have shown. Obviously, we start with sequence. And the first thing we do is we search for potential homologs or structural analogs. Since RNA sequences are based on 4 letters, and the database of RNA sequences is much smaller, it's much harder to get alignments for RNA sequences, and it's much harder to get deep alignments and good alignments. So these are usually more sparse, but whenever we get them, we use multiple sequence alignments. We make secondary structure predictions using a collection of different tools in our meta server for RNA secondary structure prediction called MesSpread RNA. And of course, we search for different types of additional information in the literature. If the templates are found for fragments or for entire structures, then we build them using our comparative modeling method, Moderna, and then all the information is fed into our method for RNA 3D structure modeling and simulation SimRNA. We also have now variants that enable modeling of RNA in complex with proteins and with small molecules. We tried to use the small molecule variant for the modeling of RNA ligand complex, but then we messed up the formatting. So I think we have not submitted that prediction with the ligand correctly, but we did this. We did the prediction, and when we're trying to, we could not submit the models for the RNA protein complex because we actually participated in solving it together with the author. So we had conflict of interest. So we ended up submitting basically only RNA models. Although one of the targets actually was solved in the complex with the U1A protein, so that was a tricky one. So we modeled a protein-bound conformation. Anyway, from SimRNA, we generate a trajectory of different conformations restrained by additional information. What is important that SimRNA does not really require secondary structure restraint, so it can fold the RNA sequence and find the secondary structure by itself, either for the whole molecule or for parts of it where the secondary structure is uncertain. But obviously, secondary structure helps. Then we cluster the best scored conformations and we do all atom reconstruction. Normally, we run all atom refinement and then select the models based on scoring. This time we had a problem because the IT at our institute decided to upgrade the infrastructure. So first they destroyed our web server, so we could not submit server models. They also prevented us from accessing our cluster. So we essentially, for this CASP, we had to model mostly based on our workstations. So it was quite an interesting period, and they haven't recovered from this update until now. So let's see how this works next time. And I mean, at some stages of this analysis, we used manual judgment. So because we couldn't rescore the models at the end, the final selection of models was in some cases based on basically human judgment of the topologies. In some cases it was not, because it was not enough time. Probably most of you have already seen that slide or my presentation about SimRNA. I mean, those of you who are interested in RNA, this is it's a method that is based on a coarse-grained model, which includes 5 centers of interactions per residue, 3 for the base, 2 for the backbone, with grids on the backbone and on the side chain. The moveset are basically random conformational changes on the level of individual atoms or the entire sections of the chain. It's a Monte Carlo approach. We use either simulated annealing or multiple replica exchange Monte Carlo, and the scoring is based on a statistical potential with parameters extracted from experimentally determined structures. So pretty much like the old school methods for protein structure modeling that dominated the field many times ago, but still, I mean, it's still working for RNA. I will show a few examples of our predictions to illustrate what I think went right, what I think went wrong. And this is one of the models that was according to the ranking on the CASP website was ranked first for target 1108, which has, well, reasonable RMSD and other scores. I mean, from the perspective, you can already see that while the overall geometry is similar, hence low RMSD, there are some changes. So for instance, in our model, we mispredicted the interaction between this loop and the potential receptor here. There's no such an interaction in the experimental determined structure. We predicted that there will be some interactions here, and as a result, actually the secondary structure in the model is slightly different, and some of the other interactions are also different. So the overall contacts are similar. The MCC for canonical interactions is 0.22, but for non-canonical interactions, the MCC is only 0.42. For stacking is 0.7, all is 0.7. So it's not great. It should be better. Okay, so this is what we actually, even though the entire structure is okay, we failed with some of the details, including things that are not so good for RMSD, but for the meaning of the structure, it's like pairing here versus pairing there versus not pairing at all. So that's the weakness of this model. Despite the overall topology is very good. And yeah, here it's the U1A binding loop, which we modeled by homology, because if we assume that if there is U1A binding loop, there must be a protein there. And so this part was a little sort of modeling based on assumption. Another case, which was the modeling of the 5' part of the 5' end of the coronavirus. This is the molecule that we have actually worked on previously. We published the paper with chemical probing of the entire coronavirus RNA, and we previously already modeled this molecule independently for SARS-CoV-2 RNA and for a couple of other coronaviruses. So we had lots of results of simulations previously, maybe not exactly for this section of the structure. So here we essentially could just cut out the model from our previous simulations, and on top of this we had our own experimental data on chemical probing. So in the generation of the model, at some point we tried to incorporate this in-house and also publicly available chemical probing data that indicated which residues may be exposed to the solvent, which may be included in the helical stems. However, the model that scored best according to CASP assessment actually has some problems that were already indicated in the assessor slide. So while overall the structures seem to be very similar for this particular target, actually we messed up the model in two places. And I think my team basically has not looked at the models, but sent the ones that form the largest, like the medoid of the largest cluster or something. Because obviously here something is clearly wrong. These two residues are not within the helix, and something horrible is happening here. This is not like, I mean, if we run the refinement, or if I looked at the structure and had a chance and time to do this manually, definitely we would correct it. The geometry overall is okay, as seen from these scores, but according to the contacts, it's not so great. Again, most of the canonical pairs are predicted correctly. Non-canonical ones, MCC is only 0.55. Stacking 0.7 overall, well, not so great. Okay, you see these two regions here, which are bad. The rest was quite okay. We predicted that this arm would be moving in the structure, but we didn't know, so we were lucky to have this model close to the good structure. The final example. Well, one of the artificial designed structures, where I will start with the secondary structure prediction. Obviously, I mean, from the kind of the structure that was here, so very long helices, ideal four-way junctions, and potential G quadruplex that would hold the ligand. We assume it must be an artificially designed sequence to match certain structural pattern. And the secondary structure prediction, well, this is actually the contacts in the model, according to the model built based on predicted structures with all the kissing loop interactions and everything. So here it seems like great. Like MCC for canonical base pairs 0.99, for non-canonical base pairs 0.83, the best in all the models we generated for all the targets. Okay, so and stacking 0.88 overall, great local contacts. What about the 3D model? Well, we can mess it up even if we have great contacts predicted. This is because during the simulation, our method got the topology of four-way junctions right in this part, but not here. So here, as you see, sort of even the spatial position of these terminal elements is actually similar, leading to relatively good RMSD, but actually the local topology is very bad. The secondary structure is great. It's quite okay, just the stacking is different, leading to different helices being stacked with each other. So getting four-way junctions right, like Eric mentioned, is absolutely essential if there are four-way junctions. And it was basically too late for us to do any manual interventions at this stage, so we could submit what we had. Now, just to summarize what we learned, what we write. I mean, at this stage, we can basically predict nearly all canonical and wobble pairs in the structure, especially the ones which are continuous in helices. So we have great MCC for those. We can often predict correctly the pseudoknotted helices, and this, in particular, kissing loop interactions, which are important in artificially designed RNAs. And in general, prediction of stacking is very good within helical segments, even if there are non-canonical base pairs within such segments. And sometimes we can get topology or four-way junctions right, and coaxial stacking of helices. I mean, in general, I think more than half of the predictions we were right. But this is not enough. So what went wrong, starting from the end? Well, topology of four-way junctions and coaxial stacking of helices certainly went wrong in a couple of cases. And when this gets wrong, everything is wrong, even if the other things are okay. What is very difficult is the prediction of non-canonical pairs, which means who pairs with whom and how. So not only what are the residues that are pairing, but what is the status of their pairing. It can be predicted if they are constrained by other canonical pairs. It's super hard if they are isolated or in long range distances. So here the average MCC for non-canonical pairs for our best model, which was only 58. The best prediction was 0.83, but mostly based on short-range interactions within the loops and not long range. And successes are rare. We have some cases where we did this right, but more often than not that we were actually wrong. And prediction of stacking interactions outside of helices is really hard. In helices and in short loops, it's very easy. But then when there is long-range stacking of single residues, this is super hard. In our model, and definitely in one of these cases, what we have to improve is the bending of the helices. In our model, the helices can actually be curved if there is a restraints that forces them to bend. In reality, there is certain Kuhn length for helices, also for single-stranded RNAs, and they would rather stay straight and then break at some point. And this can be included in the future. So I believe what would be great for the field to move forward is to use new technologies, in particular deep learning, to improve the prediction of non-canonical interactions on non-canonical base pairs and stacking beyond what is easy. So I think it's quite easy to predict things that are, it's easy to predict things that are easy to predict, and then use this as a starting information to predict stuff that is hard. We started doing some deep learning, but my group has no long-term expertise in deep learning. So if any of you are interested in collaborating with us, I'd be super happy to do something together with someone who has more experience than us in doing deep learning. We have data sets, we have good knowledge of what is required to actually train, but we just don't have experience in the very implementation of the method. So this is like a cry for help, or cry for collaboration for the future. We have some ideas how this how to generate bigger data sets for training, and then how to incorporate different methods. We know that in the existing, some of the existing deep learning methods for RNA 3D structure modeling, people are actually using simRNA to assemble the models based on what is predicted from deep learning. So at the stage of kind of refinement, and I believe simRNA is very good for modeling with restraints. So I think this can stay as a useful tool for the future. I'd like to thank the people who have been involved and the funders, and some of our software is available here. I hope I still have maybe 1 minute to answer some questions.
Thank you very much, Janusz. I have a quick question related to solvation, because it looks like some of these non-canonical interactions could be fixed if you add the solvation term. Would that be the case?
I'm not so sure. I think what RNA does, it likes to stack. Everything needs to be stacked if it can, and then it wants to pair. If some stacking prevents the pairing, the residue is pushed out to the solvent, and then if in its vicinity there is something else it can pair with, it will pair. So that's more about the interplay between the stacking and strong pairing and then whatever is left there, then you look for neighbors in 3D space.
Okay, so my question is about thinking about the larger context when you're modeling. So specifically your R1108. So you thought about the U1A region. Did you also think about the fact that it was a dimer? And if you didn't, do you think your models would have been better? Because it was in that loop that you were saying you got wrong. Right?
Yeah, I think so. There are two targets, and one of them was a dimer, and I think we tried to build a dimer for one of these these targets, and we were thinking of what could be the dimerization. I think there could be like a domain, either just stacking of the blunt helical ends with each other, or the domain swap with one of the termini. We submitted these models, but apparently none of, I mean, I haven't seen any of them scored relatively high. So maybe I misinterpreted what is presented on the website. Maybe we can talk during the break.
So regarding the outlook for using deep learning, and the main problem is to predict the non-canonical base pairing and forward junction kissing loops, something like that. So the question is, is the structural database, I mean the PDBs, enough so you basically try to get into searching all possible ones, all homology ones, and predict the right one? Or do you think there are other structures out there that are not existing in the database yet?
So I think for predicting some of the key motifs or arrangements, there is sufficient data among the structures, but this has to be extracted very carefully. There are many issues with the existing RNA 3D structures. In particular, there are modified residues. There are chain breaks. There's lots of stuff. So my lab has worked for 2 years on sort of cleaning the database. So we believe we have a data set of sort of cleaned and well-behaving RNA structures that could be used, and these can be also used as templates for modeling. I think Riju with collaborators did something like that for some of the RNAs for their deep learning, and some of the motifs can be sort of, I mean, while the templates in terms of the 3D structure would stay the same, the sequence context would change. And here, what can be taken into account explicitly by doing the modeling and using the structures for the database would essentially fulfill the issues of isostericity that Eric have emphasized for a very, very long time. And this could be isostericity at the level of individual pairs, but also isostericity at the level of entire motifs, where there's some junction where something gets in and something happens inside, but it's irrelevant. What is important is what are the angles at which the arms are protruding out of the given motif.
Okay, last question from Shoshana.
I had a general question for all the predictors. I was wondering, Ron Dror published not long ago a nice scoring function for RNA structures. Has this been used by anyone to try to see whether it's efficient to score these structures, to distinguish between correct and incorrect models?
I'm the one on stage, so sorry for being the first to answer this question, but maybe others should do it as well. What we tried to take different scoring functions from different programs and compare their utility. I don't think we have looked at I don't remember the result of Ron Dror's function, but different scoring functions from different programs, they do have some discriminatory power, but it's not very high. And this is very specific. Actually, it works very differently with models generated by different methods. The different modeling methods have different peculiarities. They generate different errors. So like Riju's method generates different kind of clashes than our method. And this is enough to actually make it difficult for different scoring functions. They are too fine-grained in a way. The models we are generating are really coarse-grained at this stage. They are like at the level, the best ones at the level of 5 angstrom, the good ones at the level of 1 nanometer, okay? And most of the scoring functions are actually more fine-grained than that. So I believe we need actually both the fine-grained scoring functions, but better coarse-grained scoring functions, which we don't have yet.